Welcome back everyone. I am Saptaparni from BioLive. Hope all of you are doing very well. So in today's video we are going to discuss about genetic cross in Drosophila, and today we are mainly focusing on Carli mutation, which is a wing mutation in Drosophila. In my previous two videos I have discussed about vestigial mutation, which was also a wing mutation, and eye color mutations like white eye color, apricot eye color, coral eye color, okay, those in Drosophila. So if you want to watch those videos, I will give the link below in the description box, and I will also add it at the end of the video. Okay, you may check it out. So what is a Carli mutation in Drosophila? See, this is a Drosophila and it is its wild type wing. Okay, when Carli mutation occurs in Drosophila, the wing becomes curled backwardly. Okay, so that is why this mutation is known as Carli mutation. So this is the Carli wing in Drosophila. Now you have to remember two things about Carli mutation. First of all, the Carli gene is located on the second chromosome of Drosophila. That means that it is an autosomal mutation. Okay. And another thing very important to remember is that phenotypically, okay, when you will get the phenotype, you will see that Carli allele, okay, or the mutant allele, which is denoted as CY, is dominant over wild type of wing allele, which is annotated as CY plus. That is why it is known as autosomal dominant mutation. Till now, whatever mutation we have discussed, like vestigial mutation, white eye color mutation, or apricot eye color mutation, we have seen that mutant allele was recessive to the wild type of allele. But here for the first time, we are getting that mutant allele, that is the Carli allele, is dominant over wild type of wing allele. Okay, got it? So from this it is clear that Carli allele, that is denoted as CY, is dominant over wild type of allele, that is CY plus. Okay, so from this we can easily say that as the Carli is a dominant allele, it will show Carli wing phenotypically in both homozygous condition and heterozygous condition. Okay, so in CY condition it should show Carli wing, and in CY plus or CY plus CY condition it should also show Carli wing. But here you can see we have written another term, lethal. That means though this genotype must show Carli wing, but the progeny is getting dead here. Okay, now why is it getting dead? We will learn about that later. Okay, now another thing is that as the CY plus allele, as it is a wild type of allele, so it will only show its phenotype in homozygous condition. Okay, because it is the recessive allele, so recessive allele only shows its phenotype in homozygous condition. So CY plus will show wild type phenotype. Okay, got it? Now second thing that you must remember is that Carli is a recessive lethal mutation. Though phenotypically Carli is a dominant mutation or autosomal dominant mutation, but it is actually recessive in its lethality. Okay, so whenever we are getting the term recessive, that means it will show lethality when it will be present in both the second chromosome. That means when it will be present as homozygous, it will show lethality. So when both the second chromosome carries the mutant allele CY, it will show death of the progeny, or we can say it as lethal. That is why though phenotypically CY must show the Carli wing, but due to its recessive lethality it is getting dead, so it is not showing any kind of wing phenotype. Okay, got it? And on the other hand, obviously in heterozygous condition, CY plus or CY plus CY, or in homozygous recessive condition CY plus all these genotype will survive. Okay, all these genotype and their respective progenies will survive. Okay, because it is recessive lethal. So whenever it will be present in heterozygous condition, it will not cause lethality. Okay, so they will survive, and obviously it will survive because it is the wild type of allele which is not lethal. Okay, so let's get into some problems. We will discuss two types of problem, one with monohybrid cross ratio and another one with test cross ratio. Okay, so the first question, see here, we have given some data of the F1 generation progenies. Okay, and you have to derive the P generation cross from here. You can see in F1 generation it is given that there are 30 curly male, 30 curly female, 15 normal male and 15 normal female. So first of all, whenever you are getting this kind of question, you must derive two things. What are those? First is sex ratio and another one is phenotypic ratio. So what will be the sex ratio here? Male is to female. How many males are here? 30. Sorry, how many males are here? 30 plus 15, that means 45. And how many females are here? 30 plus 15, 45. So the sex ratio is 1:1. Okay. Now what will be the phenotypic ratio? The curly is to normal or wild type. The curly will be 30 plus 30, 60, and normal or wild type will be 15 plus 15, 30. So it will be 2:1. Okay, so the phenotypic ratio will be 2:1. Now I have written the 2:1 as monohybrid cross ratio. Why? You may ask me that 3:1 is known as monohybrid ratio, right? So why 2:1? Because 3:1 is the monohybrid ratio in Mendelian process. But whenever we are talking about lethal alleles, then the monohybrid ratio becomes 2:1 instead of 3:1. Okay, so you have to remember it that the monohybrid cross ratio for recessive lethal alleles is 2:1, not 3:1. Okay. So from this it is clear that it will be a monohybrid cross. Now next thing that you must always remember, that whenever you will get any kind of monohybrid cross, you have to take both parents as heterozygotes. Then only you will get all types of progeny that are given in the question. Okay. So we are taking male and female both as heterozygotes. So see here, this is the female of P generation and this is the male of P generation. So we are taking female as heterozygote. So one autosome of the female will be CY and another one will carry CY plus. And obviously as these alleles, curly alleles are present on second chromosome, we have to also denote the X and Y chromosomes here. Okay, for female there are only X chromosomes, so both the X chromosomes are wild type in nature, so plus plus. And in case of male, we are also taking the heterozygous condition because it is a monohybrid cross, so CY plus, and as it is a male, there is a wild type X chromosome and wild type Y chromosome. Okay, now we are deriving the gametes. So what will be gametes here? One will be CY and plus, or CY and X chromosome, and another one will be CY plus and plus, or CY plus and X chromosome. Okay, and from male we will get four types of alleles. One will be CY and X chromosome, you can see CY and X chromosome. Another one will be CY and Y chromosome, CY and Y chromosome. Then one will be CY and X chromosome, sorry, CY plus and X chromosome, CY plus and X chromosome, and another one will be CY plus and Y chromosome, CY plus and Y chromosome. Okay. So when we will write it in the checker box, we will get checkerboard, we will get the cross of F1 generation. Okay. The progenies of F1 generation. We are writing male upward and females lateral side. Okay, so these are the four gametes of male and these are the two gametes of female that we have derived. Okay, so after crossing we will get CY plus plus. Now I have told you, whenever as CY is the recessive lethal allele, when both the autosomes will carry CY allele, it will be dead. Okay, so it is lethal. So this progeny will not survive. Okay. Next, CY plus X and Y chromosome. So as this is also lethal allele in homozygous condition, so it will also get dead. Okay, so it is also lethal. Now here cy plus plus, as here one cy plus and one cy is present, so obviously Carly is dominant over wild type. So here in heterozygous condition it will show Carly mutation, and here as there are two X chromosome it will be Carly female. Now similarly here you can also see cy plus cy and X and Y chromosome, so it will be Carly male. Okay. Now here similarly cy plus plus, so it is Carly female, cy plus X and Y, so it is Carly male. Here you can see both the autosomes are carrying cy plus alleles, that is wild type of allele, and we know that wild type is recessive, so in homozygous condition it will show its phenotype as wild type. Okay, so and here two X chromosomes are present, so it is wild type female. And here also you can see cy plus cy, so it is wild type, and X and Y chromosome, so male. Okay, so from here we can get that how many Carly males are there, how many Carly females are there, how many normal or wild type males are there, and how many normal or wild type females are there. How many Carly males? One Carly male, two Carly male. Okay, then how many Carly female? One Carly female, second Carly female, so two Carly females. How many normal or wild type males? Only one wild type or normal male. And how many wild type female? Only one wild type or normal female. So our ratio 2:1:1 is achieved here. Okay, so see here if we get the ratio, we will get 1:1:2, right? The phenotypic ratio will be this. Okay, so the phenotypic ratio of the given question or of the given F1 progeny is being proved by our cross. So our P generation cross is correct here. Okay, got it? Now let's discuss about the second question. Here you will see that it is a test cross question. So here also the phenotype of F1 generation progenies are given and their numbers are also given, and you have to derive the P generation cross. Okay, so you can see all Carly male, female, wild type male, female are same in number 10, 10, 10, 10. Okay, so first of all we will derive the sex ratio. The sex ratio will be obviously male is to female equal to 1:1. Okay, because how many males are there? 10 plus 10, 20, and how many females are there? 10 plus 10, 20. So it is 1:1. Now the phenotypic ratio Carly is to wild will be how many Carly are there? 10 plus 10, 20, and how many wild types are there? 10 plus 10, 20. So it is also 1:1. Now we know that 1:1 is the test cross ratio. Okay. So in case of test cross, we know that one parent should be homozygous recessive in nature. Okay, so here we can do two crosses. One is the simple cross and another one is the reciprocal cross. In the first cross we will take one parent as homozygous recessive, because it is the rule or it is the characteristics of any test cross. So here we are taking female as the homozygous recessive. So female has cy plus or recessive allele on both the autosomes. Okay, and as it is a female, obviously two X chromosomes are there which are wild type in nature. Now obviously if you are taking one as the homozygous recessive, then the male or the male parent should be like heterozygote. Okay, so it should carry cy plus cy and X and Y chromosome, and if we don't take it as heterozygote we will not get the desired F1 progenies that are given in the question. Okay, so how many gametes should be formed from here? From female we can get only one type of gamete, that is cy plus and X chromosome. So here only one type of gamete could be here, cy plus and X chromosome. And from male we can get four types of allele. One is cy plus and X chromosome. Here you can see cy plus and X chromosome. Another should be cy plus and Y chromosome, cy plus and Y chromosome. Then one should be cy and X chromosome, cy and X chromosome. Then cy and Y chromosome, cy and Y chromosome. Okay, and when we will do the checkerboard we will get that one progeny will be cy plus plus. When both the autosome carries cy plus or wild type allele, then recessive allele, then the phenotype should be wild type and female. Okay, Here also CY plus, that means both the autosomes are carrying recessive alleles, so the phenotype should be wild type, and as there are X and Y chromosomes, so wild type male. Here you can see CY plus and CY, so it is curly in nature because curly is dominant over wild type allele. Okay, so it will be curly, and here two X chromosomes are there, so curly female. Here also CY plus CY is dominant over CY plus, so the phenotype should be curly, and as there are X chromosome and Y chromosome, it should be curly male. So what is the ratio here? Curly male is to female is to wild type male is to female. How many curly males are there? 1. How many curly females are there? 1. Okay, how many wild type males are there? 1. And how many wild type females are there? Only 1. So the ratio is 1:1:1:1. Okay, which is given in your question. See here, the ratio should be 10:10:10:10. So 1:1:1:1. So the ratio is proved. So the cross is correct. Okay. Now we can also do the reciprocal cross, and from this we will also get the same result. So what is reciprocal cross? I have told you that as it is a test cross, so obviously there should be one parent which should be homozygous recessive. Here we have taken female as the homozygous recessive. Okay, so in the reciprocal cross we will take the dad or the father as the homozygous recessive. So here we have taken father as the homozygous recessive. So both the autosomes of the father is carrying CY plus allele or recessive alleles, and one X chromosome on one Y chromosome. And female should be, then what should be the genotype of the mother or the female? It should carry heterozygous alleles in its second chromosome. So one chromosome is carrying CY plus, another one is carrying CY, and both the X chromosomes are here. Okay. So how many gametes should be formed from the female? From the female there should be 4 gametes. One is CY plus and X chromosome, and another one should be CY and X chromosome. So CY and X chromosome. Okay. And from male also there are 2 types of gametes, CY plus and X chromosome, and another one should be CY plus and Y chromosome. CY plus and Y chromosome. So when we will do the cross here, putting male gametes on the upside and female gametes on the lateral side, we will get CY plus plus, as both the autosomes are carrying recessive alleles, so it will be wild female. Then CY plus X and Y, so it is a male, and as both the autosomes are carrying CY plus alleles, so it would show wild type of phenotype. Then CY plus, so as it has 2 X chromosome, it is female, and as it has one CY allele which is dominant over CY plus, so it will be curly female. Okay. Here CY plus X and Y chromosome, as it has X and Y chromosome, so it is a male, and as it has one CY allele which is dominant over CY plus or wild type allele, so it will be curly male. Okay, so from this also we can see that how many curly males are there? Only one curly male. How many curly females are there? Only one curly female. How many wild type of males are there? Only one wild type of male. And how many wild type of female are there? Only one. So the ratio is also proven by this reciprocal cross. Okay, so in case of test cross, you can take any one parent as homozygous recessive. It could be either mother or father. Okay, you can take anyone as homozygous recessive, and another parent should be heterozygous in nature. Okay, and then the cross will match the F1 progeny given in the question. Okay, so this is all about today's video. In my next video, we will discuss about lethality when the sex ratio will not be 1:1. Okay, when the sex ratio will be either 2:1 or 1:2, then how the crosses will change, we will see in those videos. Okay, so with this, I end my today's video here. If you find this video useful, don't forget to hit the like button and share it with your friends. And don't forget to subscribe to my channel for more such videos. Thanks for watching and happy learning.